Let's review the first semester of AP Bio in about
20
minutes. There's not a lot of time for formalities today because we have a ton of topics to cover from units 1 through 4. So let's begin right away with unit 1. Unit 1 is on the chemistry of life, and this unit starts with water. So the thing that you have to know about water is that water has a chemical formula of H2O, which means that it has 1 part oxygen and 2 hydrogens in a bend shape. But a hydrogen atom only has 1 proton in its nucleus, meaning that against the 8 protons of the oxygen, the central molecule, these electrons are going to be clustered more towards the oxygen side, creating a slight negativity on the oxygen end and slight positivity on the proton end. And this is what we call the polarity of water. And water being polar gives rise to 4 main properties that you have to know. These are adhesion, cohesion, and surface tension, high specific heat leading to water's ability to moderate temperature, water's strange ability to decrease in its density as it freezes, and its capability as a universal solvent. Of course, each of these properties have to be connected to something biological. For example, adhesion and cohesion are often demonstrated with the ability of trees to perform transpiration as water molecules stick to the sides of the xylem as it pulls more water molecules from beneath in order to transport it from the ground all the way to the atmosphere. Water's high specific heat also leads to the stability of the temperature of water, allowing organisms to thrive. And the decreasing density of water, of course, allows ice to float above liquid water, meaning that a thin layer of ice can act as insulation on top of small bodies of water, allowing organisms to thrive over periods of freezing temperatures in the wintertime. But the last one is the most important one, which is water's ability to act as a universal solvent. So here we're going to learn a few words: hydrophobic and hydrophilic. Hydrophobic refers to molecules or substances that do not dissolve in water because they have no polarity. It's kind of like trying to stick a magnet against the plastic. Water is polar, so it's kind of magnetic, and of course the plastic non-polar substance will not react with the magnet. However, hydrophilic refers to anything that does dissolve in water, so these include things like sodium chloride, which can dissolve into sodium ions and chloride ions, which are positive and negatively charged, and of course those are going to react with the polarity of water, and even larger proteins like lysozyme, which happens to have a bunch of polar groups that are associated with it. Next, we move on to the elements of life, and the only thing that you have to pay attention to here are the elements that comprise the 4 major macromolecule classes that we're going to see later, plus a few more. But here they are. We have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. That set is the one that you have to know. And the other thing to associate with these elements is that they are relatively common within our universe, meaning that life is pretty opportunistic, using whatever on hand in order to create life. But before we get to macromolecules, we have a subunit on carbon chemistry, and there are a few things to cover here too. So first, we need to understand that carbon is the atom that acts as the backbone of most biological molecules. And the reason for this is that carbon has 4 valence electrons, each of which can be used to form a covalent bond that can create complexities such as length, double bonds, triple bonds, branching, as well as creating a bunch of molecules that have the same chemical formula but different structures. And these are going to be called isomers and enantiomers. Isomers break down into structural and geometric isomers, while enantiomers, as you can see on the screen, are simply mirror images of molecules. This is incredibly important. It's the first time that we're introduced to the concept of structure and function. While these molecules may have the same formula, because they have different structures, they're going to perform different functions. So the idea of structure correlating to its function is going to be pivotal from this point moving forward. And now we get to the structure and properties of macromolecules and nucleic acids. All macromolecules except lipids are polymers, which means that they're built with monomer subunits that elongate into a long chain. The process of creating polymers is called polymerization reaction, and these are dehydration syntheses, meaning that a water molecule is released every time a monomer attaches to another monomer or a growing chain of polymer. The reverse can also happen, and that reverse is going to be called hydrolysis, a term that you'll probably see a lot in the subsequent chapters. In AP Bio, we need to know about 4 macromolecules. We have carbohydrates, proteins, lipids, and nucleic acids. Carbohydrate monomers are called monosaccharides. Their polymers are called polysaccharides. The most common monosaccharide that you have to know is going to be C6H12O6, glucose. This is a hexose, meaning that it has 6 carbons, and it's the most important one that you'll see within the first half of AP Bio. When a bunch of monosaccharides get together, we call these bonds glycosidic linkages. And in the case of polysaccharides, we have to know about 3. We have glycogen and starch, which are storage polysaccharides for animals and plants respectively, and we have cellulose, which is a structural polysaccharide for plants. What's important to note is that the only difference between starch and cellulose are the type of glucose they use. So starch uses alpha glucose monomers, while cellulose uses beta glucose monomers. And because the bonds are different, certain enzymes like amylase that can break down starch will not be able to break down cellulose. And this is one of the reasons that we learned that herbivores, like cows for instance, have special microbes that can break down cellulose through enzymes such as cellulase, forming a symbiotic relationship of mutualism. Moving on to lipids, lipids are not true polymers, meaning that they do not elongate indefinitely. However, we have to learn about 3 different types of lipids. We have fats/oils, we have phospholipids, and we have steroids. Now, steroids are going to come into play a little bit later in chapter 11 or cell communication, so we can put that aside for right now. But what's important is that fats and oils are composed of a glycerol molecule attached to 3 fatty acid chains, which can either be straight, as in saturated fatty acids, or kinked in unsaturated fatty acids. But do keep in mind that this unsaturated versus saturated fat can play a role in membrane fluidity that we'll talk about in a few moments. Phospholipids is probably the star of this section, and that's because phospholipids are amphipathic, meaning that they are both hydrophobic and hydrophilic simultaneously. Now, glycerol molecule still plays a role here, but only 2 fatty acid chains are found on one side, while a phosphate molecule, which is charged negatively, is going to be found on the other side. This means that the negative phosphate head can dissolve in water, while the lipid tails do not dissolve in water. Did I forget to mention that lipids are nonpolar and they don't dissolve in water? Sorry about that. Next, we have proteins. Now, proteins probably the most important macromolecule that we have in AP Bio, and they're comprised of monomers that we call amino acids. When they're polymerized, they're going to be called polypeptides because they form peptide bonds in between each amino acid. A good starting point to studying proteins is what these amino acids look like. There are 20 of them, and they are categorized into polar and nonpolar, charged, and charged can be subdivided into negative and positive, the bases and acids. But what differentiates each amino acid from each other is the presence of the R group. The R group is a chemical signature that each amino acid has. Now, when we form a long chain of amino acids, you could imagine a crazy number of possibilities in terms of sequence, and that sequence would place these R groups next to each other, which then allows them to chemically react, folding that strand of polypeptide into a 3-dimensional structure. And speaking of structures, we have to know that the primary structure is simply the sequence of those amino acids. Secondary structures are alpha helices and beta pleated sheets, which are commonly repeating themes in the folding of these amino acids. And tertiary structure is sort of that unique structure that you see due to all of the interactions found between the R groups. Quaternary structures do exist in certain proteins when multiple polypeptides come together to form a singular functional protein. Lastly, we have nucleic acids, but at this point in the game, nucleic acids aren't going to be as important. However, we do have to know the structure, so I'm going to show you a picture right here. Nucleic acids are formed from monomers called nucleotides. They bind to each other through a bond called phosphodiester bond. Each nucleotide is composed of a nitrogenous base, a 5-carbon sugar, and a phosphate group. These bases can differ between DNA and RNA. DNA has adenine, thymine, guanine, and cytosine, while RNA has adenine, uracil, guanine, and cytosine. The 5-carbon sugar is slightly different as well. The sugar on the DNA, which is a deoxyribose sugar, is missing that oxygen from the 2nd carbon. The superstructure can differ as well. RNA is typically single-stranded, although they can fold into different shapes later, as we'll see. But DNA is definitely an anti-parallel double helix structure. Anti-parallel meaning that one of the DNA strands will go from 5 to 3' from top down, and the other will go from 5 to 3' from bottom up. These primes, of course, refer to the ends of the DNA molecules, which are represented by either the phosphate or the hydroxide group attached to the 5' carbon and the 3' carbon as we find them on that deoxyribose or ribose sugar. And that completes Unit 1. Now on to Unit 2 on cell structure and function. So this unit starts off with the 3 components of the cell theory, and those are that all living things are made of cells, the cell is the basic unit of life, and all cells come from pre-existing cells. Just the 3 facts to remember. Now, we also get a sense that cells are really, really small, which means that we have to study them with microscopy. Remember that there are light microscopes and electron microscopes, so please note that electron microscopes, while they're really good at resolving fine details, must work with dead samples. Now, the second question is, why are cells really small? And the answer to that seems to be the surface area to volume ratio. And this simply means that as cells get bigger, the surface area that allows for exchange of materials in and out of cell becomes smaller and smaller and smaller relative to the volume that it must support. And this means that cells can only get so big before the surface area gets too small for the volume that it must sustain. You also should take note of the differences between animals and plant cells. For example, plants have a cell wall and they have chloroplasts while the animal cells don't. But there are more similarities and differences, and you should take note of the organelles and see what they do, but that's not the most important part for AP biology, because for AP bio, what they want you to know is how the endomembrane system is all interconnected through the merger and the disjoining of the phospholipid bilayer. So from the nucleus to the endoplasmic reticulum, both smooth and rough, and the Golgi apparatus and the vesicles that continuously exchange materials between them, all the way to the plasma membranes, are a single continuous system that we call the endomembrane system. Do note that mitochondria and chloroplasts are not part of the endomembrane system because they're believed to have originated from prokaryotic origins, as evidenced by the double membrane as well as having their own DNA. Then we get to focus a little bit more on the cell membrane. Remember, the plasma membrane is comprised of phospholipid bilayers. The first thing that we learn is something called the fluid mosaic model, which indicates that phospholipids are not really the only thing that make up the plasma membrane. We have integrated proteins, peripheral proteins, glycoproteins, glycolipids, and even cholesterol that contribute to the fluidity and the mosaicism that is the plasma membrane. The fluidity can be controlled by a couple of factors. The presence of cholesterol seems to inhibit the cells from getting too fluid or too rigid, while having more phospholipids with unsaturated tails seem to create additional fluidity as a result of the spacing between the phospholipids, while having more saturated fatty acids tend to pack them a little bit tighter, resulting in a more rigid plasma membrane. Now, the plasma membrane provides a relatively impermeable surface through which polar or charged substances cannot pass. This is, of course, because the lipid tails, which is a nonpolar region, does not allow charged or polar things to pass through. However, in order to allow for certain things that cells do want to pass, cells have added things called channel proteins, and this is where we're introduced to the idea of facilitated diffusion. Now, of course, diffusion is the action of materials moving from an area of high concentration to low concentration, and facilitated diffusion simply allows a channel or a carrier protein through which materials can simply move from an area of high to low. Now, note that these channel proteins can actually be closed or open, so it's not like it's completely unregulated. Next, if you wanted to move things from an area of low to high concentration, then you can do so by using a process called active transport. The proteins that does this are generally called pumps, and it usually uses ATP in order to drive this energy-hungry process. Now, the last thing to note about the membrane exchange is the exchange of water. Now, the water is actually allowed to move freely between in and out of the cell on account of the presence of something called aquaporins. Aquaporins are channel proteins like we saw before, but are very specific to water. But we can't ask the question of what concentration of water is going to be higher or lower, because concentration itself has water as the denominator in the equation. So what happens with water is that it moves in accordance to what we call osmosis. Osmosis is the action of water moving from an area where there's lower solute concentration to an area where there's higher solute concentration. Unlike diffusion, though, we're not looking at a particular solute, but the totality of all the solutes that we measure in osmolarity. And it happens to move water in the opposite direction to the movement of solutes if they were possible. So for instance, water will move from an area where there's lower solute concentration to where there's greater solute concentration. The way that I like to think about this is that water is always moving to dilute something. And here we're introduced to the idea of isotonic, hypotonic, and hypertonic solutions. Now, solutions is always referring to the outside of the cell. So in a hypertonic solution, there are more solutes on the outside, meaning that water is going to move from the cell to the outside, shriveling the cell. Or in the case of plants, plasmalizing it. Isotonic, iso meaning the same, would actually result in the net movement of water being equal in and out, so we're all good there. But in hypotonic solutions, where the surrounding has lower solute concentration, you should expect water to come rushing into the cell, leading to lysis in animal cells, or simply being turgid in plant cells. And they're perfectly happy with this because the cell wall can withstand that pressure. And one last little bit from this unit is about cell compartmentalization. Here, we need to understand that unlike prokaryotic cells, eukaryotic cells have organelles. And the idea is that these organelles allow for very specific conditions within them in order to facilitate enzymatic reactions that operate best at certain pHs or certain solute concentrations. So that obviously provides benefit for eukaryotic cells who can carry out multiple enzymatic functions in their optimality across many organelles that they have. Unit 3 begins with enzymes and works through photosynthesis and cell respiration. Now, enzymes are proteins that have the ability to catalyze chemical reactions, and they do so by having a very particular shape that can bind to substrates and change the structure of the substrate in a way that reduces the amount of energy required for that substrate to undergo a chemical change. And this applies to reactions that both require energy or does not require energy, otherwise known as spontaneous reactions. The things to keep in mind here is that enzymes are affected by environmental factors such as pH, temperature, and salinity. Now, the temperature is probably the easiest one to understand. As temperature increases, the enzyme can lose its shape, meaning denaturation, and the loss of that shape would of course affect the active site, resulting in a reduced or a non-functional protein. You should also note that enzymes are not used up during a chemical reaction, so as long as substrates are available, you will get that enzyme to function over and over again. You also learn a little bit about something called allosteric regulation. This means that we have a negative feedback system in which enzymes can be turned on or turned off by the amount of products that you have. So the more products you have, you want to slow down that reaction. That product might bind to that enzyme at a different site than the active site, changing the shape of of the active site, disallowing the substrate to bind. This allows for a negative feedback loop, which then allows for a particular rate of reaction to be maintained. Now, we talk about enzymes because there are many enzymes that are involved in the photosynthetic process. Photosynthesis is the means through which energy from sunlight can be harnessed in order to produce glucose from inorganic carbon dioxide and water. And here's how it's going to work. Plants can absorb energy of light through pigments that we call chlorophyll and carotenoids. These pigments contain electrons that can be excited, and that's a technical term, by the way, by a photon of a very particular wavelength. Now, for chlorophyll, this is going to be in the blues and the reds, which is why it also happens to reflect or transmit green. These chlorophyll are found in complex proteins called photosystems within the thylakoid membrane. These photosystems contain many chlorophyll molecules which can pass their electrons through what we call the electron transport chain. As the electron transport chain harnesses the energy of that excited electron, it starts to pump hydrogen ions or protons from the stroma into the thylakoid inner space. This creates a concentration gradient of hydrogen ions which want to leave, and the only way they can leave is by the ATP synthase, and as they leave, they alleviate that pressure gradient, creating ATP. The electrons are once again excited in photosystem 1, where they're passed on to an electron acceptor that we call NADPH. At the same time, loss of these electrons from this linear process are replenished by water. Now, the ATP and NADPH that were produced by the aforementioned reaction that we call the light reaction is used in the Calvin cycle. The Calvin cycle has 3 stages: the carbon fixation, reduction, and regeneration. During the fixation stage, RUBP and a carbon dioxide molecule are bound together via an enzyme called ribisco into forming a 6-carbon intermediate, which then splits right into 3-PG. Into the 3-PG, we use those ATP to upgrade their energy into 1,3-bisphosphoglycerate, which is then reduced by NADPH with those electrons to form glyceraldehyde 3-phosphate. Some of these glyceraldehyde 3-phosphates are used in the production of sugar, while the rest are regenerated back into RUBP. Now, this is the typical way that photosynthesis proceeds in C3 plants. Notice that in C4 and CAM plants, which live in arid areas, they have special adaptations that can deal with this problem. But for that video, click the link below. Now that we've produced the glucose or the sugar molecules, this can be broken down through the process of cell respiration. This happens in plants, but it also happens in animals who consume those plants. The process of breaking down that sugar or cell respiration is largely broken down into 4 separate steps. We have glycolysis, pyruvate oxidation, Krebs cycle, and oxidative phosphorylation. In glycolysis, which is a multi-step process involving many enzymes, that glucose is broken down into 2 halves, which each are called pyruvate or pyruvic acid. During this process, you actually have to invest 2 ATPs, but you get 4 back, so you have a net gain of 2, and you also get to break down some bonds, which releases those high energy electrons that are holding glucose together, meaning that you still have NADH, which are electron carriers used in cell respiration. Next, that pyruvate is imported into the mitochondria, where it loses a carbon dioxide, but instead gains a coenzyme A, becoming acetyl-CoA. During this process, another bond is broken, so electrons are released. You get the formation of an NADH molecule. Acetyl-CoA can now enter the citric acid cycle, where it first joins an oxaloacetate, forming a 6-carbon acid called a citrate, which is why it's also called citric acid cycle, and it undergoes several chemical reactions, releasing 2 more carbon dioxides, forming 3 NADH, 1 FADH2, and even an ATP. Now, if you count per glucose, this would be 6 NADH, 2 FADH2, and 2 ATP, because there were 2 pyruvic acids that went into the mitochondria prior to the Krebs cycle. This means that by this point, we've already produced 4 ATP, but remember that there are 30 to 32 ATP that could potentially be produced from glucose, and where that energy still is within these electrons being carried by NADH and FADH2. The NADH and FADH2 drop off their electrons at the protein complexes on the inner membrane of the mitochondria, and much like the ETC that we saw on the thylakoid membrane, these electrons can drive pumping of those protons, but this time from the matrix of the mitochondria into the intermembrane space. As these protons fill the intermembrane space, they want to be relieved of this chemoosmotic pressure, in which case they fall through the ATP synthase back into the mitochondrial matrix, forming a lot of ATP. This process is called oxidative phosphorylation. The electrons that arrive at the end of the electron transfer chain are removed by oxygen, the final electron acceptor, which joins with 2 hydrogen ions in order to form water. And finally, we arrive at unit 4, which is on cell communication and the cell cycle. In cell communications, we learned that cells communicate with each other using chemical signals. We get a glimpse into this world using Saccharomyces cerevisiae as examples. Now, the A cell and the alpha cells release A factors and alpha factors respectively, but can only be heard by the other cell type because the other cell types only contain those receptors that can listen into the opposite mating type signals. Now, as we expand this chapter further, we learned that Earl W. Sutherland studied epinephrine and its effects on liver cells in converting glycogen into glucose. And And using that example, let's go through this chapter very quickly. Sutherland outlined that there are three main stages to cell signal transduction. There is reception, transduction, and response. In the reception part, we learned that there are three major categories of cell surface receptors. These are G-protein coupled receptors, receptor tyrosine kinases, and ligand-gated ion channels. Focusing on G-protein coupled receptors, what happens here is that a ligand attaches to a G-protein coupled receptor, which then changes its shape in order to knock out that GDP residing on the G-protein. GTP quickly takes its place, the G-protein gets activated, it migrates down the periphery of the cell, therefore activating some other enzyme downstream in the pathway. Now, in the case of epinephrine, this other enzyme happens to be adenylyl cyclase, which, very importantly, produces a second messenger called cyclic AMP. Cyclic AMP is produced by adenylyl cyclase cleaving off two phosphates known as pyrophosphate and creating a cyclical attachment from the 5' to the 3' of that ribose sugar, creating an AMP that is cyclical in nature. Now, cyclic AMP is an important second messenger because it has this ability to activate the transduction. Because in the second part of this pathway called transduction, that second messenger activates a protein kinase. Now, kinase is a class of enzymes that can phosphorylate other downstream substrates. Now, this can activate or deactivate substances, but in the case of protein kinases, they're typically activated by cyclic AMP. Now, once the kinase is activated, they themselves activate about 10 kinases down below, and each of those kinases activate another 10 kinase below. And this is what we call amplification. And by the time we get to the bottom, we can finally get to the response by potentially phosphorylating and activating an enzyme that is supposed to carry out the function in this cytoplasmic response. Now, in the case of epinephrine, the enzyme to be activated is glycogen phosphorylase, which converts glycogen into glucose. Now, there are other types of receptors. For example, intracellular receptors for ligands such as testosterone or estrogen, which are lipid-based. Well, those signals come straight through the lipid bilayer, so they bind inside. And I just mentioned the cytoplasmic response. There's also the gene expression response. So, for instance, in a growth hormone, all of these pathways will ultimately result in the activation of a transcription factor, which can activate the transcription of a particular gene, leading to the production of a protein. And there is this tidbit on apoptosis. Remember that apoptosis is the cell's ability to self-destruct, and this usually happens as a result of a problem within the cell or the developmental pathway that necessitates the destruction of a cell. This is also driven by numerous chemical pathways within the cell, which is why it's included in the cell communication pathway, as this example shows. And now we get to the cell cycle. Remember, the cell cycle is the first time that we think about cells living across time, across generations, instead of seeing things that are happening in a static moment in time. And in the cell cycle chapter, we learned that cells begin their life at G1 of interphase, where they grow and develop into the cells that they're supposed to become. But an internal or an external signal will facilitate their transition into the S phase, which will then duplicate the DNA that is within the nucleus. And the duplication of DNA is important because while the organelles and other substances can be distributed between the two daughter cells at a haphazard way, the information that is contained within the nucleus must be perfectly copied in order to make sure that the two daughter cells has the exact same data after mitosis is complete. So once the DNA has been replicated in the S phase, we enter the G2. G2 is going to have numerous checkpoints to ensure that everything looks good, make sure that there are no errors on the DNA, and that we have sufficient energy available to complete the mitotic event. And that's sort of the end of interphase, because as soon as mitosis begins, we go into the mitotic phase, which begins with prophase, goes to prometaphase, and then the metaphase, and then the anaphase, telophase, and that overlaps a little bit with cytokinesis, which is the physical breaking apart of the two daughter cells. During prophase, the most important bits are that condensation of DNA occurs, so we start getting the formation of those chromosomes with the chromatids that are held together at the centromeres. We also get the spindle fibers forming what we call aster formations, which means that we have these microtubules that are going to be used in order to segregate those chromosomes into the two daughter cells. In the prometaphase, we have the dissolving of the nuclear membrane, and we also have the full attachments of those spindle fibers onto the chromosomes. During metaphase, those chromosomes are moved where they line up in a single file line ready for those chromatids to separate. During anaphase, those chromatids separate and move towards the mitotic poles, and during telophase is the reverse of our prophase, meaning that the nuclear envelope reforms, the chromosomes decondense, and you now almost have two daughter cells that are separate and ready to live their own lives. Cytokinesis is driven by actin filaments that form sort of a zip tie between the two daughter cells that get smaller and smaller and smaller, eventually dissociating the parent cell into two daughter cells. And there we have it, folks. I'm not sure what the timer is going to say, but the most important thing is that we've talked about primarily all the important bits from Unit 1 through Unit 4. And this video is not going to be completely comprehensive on everything that you need to know, but I have those links for Unit 1 through Unit 4 videos, as well as problem-solving videos if you want to look through those as well. And if you found this content useful, then consider subscribing to our channel and clicking the like button. It doesn't cost you a cent, but it helps us a lot. And because there's still semester 2, keep up with our videos, and hopefully we'll take you all the way to the May exam. This has been Mikey with Avo Prep Academy. See you in the next video.